Do you know that men are 4 times more likely to be diagnosed with bladder cancer than women? Bladder cancer is more prevalent in men than in women and usually affects older adults, though it can happen at any age. Cigarette smoking is the main risk factor for bladder cancer and has been reported to be responsible for half of the cases. So what is bladder cancer? It is a type of cancer arising from the tissue of the urinary bladder. Bladder cancer is a disease in which cells grow abnormally and have the potential to spread to other parts of the body. This figure illustrates the abnormal development of tissues forming tumors within the bladder. Bladder cancer varies from moderate disease with low mortality to very high-grade tumors associated with high mortality. Did you know that chemotherapy is the first treatment that is given to bladder cancer patients? Chemotherapy is delivered in cycles to cause fractional killing of non-synchronous growing cancer cells, and treatments are spread out to let the normal tissues recover between the cycles. However, drug resistance in bladder cancer cells causes repopulation of remaining cancer cells that are called chemoresistant. It is an unpleasant occurrence that reduces the effectiveness of chemotherapeutic reactions in subsequent cycles and causes the recurrence of bladder cancer. Have you ever wondered how the cells in our body work? The answer is through the help of trace elements. So, trace elements is a term that refers to chemical elements found in a relatively small quantity in living tissues. These elements are absolutely vital to the upkeep of our biological functions. While trace elements are important components in our biological processes, excessive concentration or deficiency of them can cause body health toxicity and lethal diseases such as bladder cancer, prostate cancer, and others. In the present work, we are currently studying neutron activation analysis (NAA) on trace elements in human bladder tissue. NAA is a nuclear process used to determine the concentration of elements in given material. You must be wondering, how does the NAA work, right? The basic principle is neutron captured. Neutrons fired at the sample then react with the nucleus. After receiving the neutron, it goes to an excited state that decays quickly to the lowest energy state. In that transition, it emits prompt gamma radiation that has a very high energetic range. After that stage, the nucleus can be a stable nucleus again, or it can be a radioactive one. And NAA works with radioactive nuclides. Usually, the radioactive nucleus decays to the beta- particles and a cascade of gamma rays are afterwards also emitted, which is called as delayed gamma rays. NAA works with delayed gamma rays. Information we gain from this gamma radiation is the energy of the gamma ray that are the characteristic for each element. That means, through the energy, we can determine the element inside the sample, and the intensity of these gamma rays are also proportional to the amount of element inside the sample. This project utilized NAA, which is available in the Nuclear Malaysia Agency that is located in Bangi. The only nuclear research reactor in Malaysia, Nuclear Malaysia Trigger Mark II reactor, is the neutron source for this NAA work. The reactor operated at 750 kW with a thermal neutron flux of 2 × 10 to the power of 12 per centimeter squared per second. This present work examined 10 bladder tissue samples obtained through a mastectomy provided by the Department of Pathology, Faculty of Medicine from University of Malaya, and all of them obtained after the patients undergo chemotherapy. Before sending the samples to the Nuclear Malaysia Agency, additional preparation samples need to be done as the number of samples are little, so they cannot replicate and need to re-irradiate the samples to get the elements of longer half-life, such as drying the samples, grinding and pulverization of the samples by using mortar pestle, store the samples, which is in dust size, inside capsules and label accordingly. Then, weight each sample and lastly record each weight of the samples and label accordingly. Okay, then the samples are ready to be irradiated by the NAA. So, have you ever seen the ways to analyze the samples by using NAA? I don't think so, right? So, I'm here to show you exactly the steps to analyze the samples by using NAA. From the results obtained, there are 3 elements that were found to have high concentration in the bladder tissue samples, which are calcium, magnesium, and sodium. According to past research, the concentration of magnesium, calcium, and sodium were found in the cancerous tissue. According to a finding from Abdul Gawad in 2016, it stated that high concentrations of calcium and magnesium were observed in grade 1 and grade 3 bladder carcinoma. Accordingly, heavy metals trace elements concentration in bladder cancer tissue possibly affects the histopathological cancer type as well as the grade of the cancer. Also, the researchers stated that the possibility of rupture of tumor surface and leakage of the heavy elements such as calcium and magnesium in the urine could be diluted and affect the tissue concentration. Malaysia has a variety of food, and even without realizing it, there is always some hidden salt content in those food. 5 grams of salt equates to 2,000 mg of sodium, which may be the reason why the result we obtained shows a high concentration of sodium in the bladder tissue. So, based on this data, Malaysia can build up a database system at the nuclear cancer research. Did you know that this is the first study about neutron activation analysis NAA on trace elements in human bladder tissue that is conducted in Malaysia?